So today I'm going to kind of give sort of a high level overview talk of some of the methods that we've developed for statistical brain network analysis, and then also discuss or touch on some potential future directions we may take. So this will be sort of a higher level talk and not like a detailed single paper talk. And please feel free to interrupt me at any time with any questions or comments or points of clarification. I've left plenty of time for that. So first, I'd like to acknowledge my co-authors on the relevant work for this talk. Chad Tomlinson was a PhD student of mine. He was at Chapel Hill. I have an adjunct appointment there, so I co-advised him in the biostat department. And Mohsen Barami was actually a PhD student of mine at Wake in biomedical engineering. He's now on faculty with me at Wake. Heather Schoppel is a biostatistics colleague of mine. And then Paul Laurenti is sort of my main longtime neuroimaging collaborator. And we're all part of this laboratory for complex brain networks. And I'd also like to acknowledge the funding that facilitated some of this work. So here's an outline for my talk. First, I'll talk about sort of the impetus for brain network analysis more generally. I'll then get into descriptive methods. So once you have these brain networks, how do you describe them? And I'll briefly touch on this issue that these network metrics or descriptive metrics that we calculate are actually random variables, but we typically ignore that in the field. And so this is a sort of area of needed research. And then I'll dive into the meat of the talk, which will talk about sort of three complementary frameworks that we've developed for brain network analysis: mixed modeling framework, a distance regression framework, and a hidden semi-Markov modeling framework. So now connectivity and network analyses have exploded over about the last 21 years or so now. And they've already shown a lot of potential into helping us understand normal and abnormal brain function. Now, functional connectivity analysis examines associations between time series in specific regions of the brain, whereas network analysis quantifies associations between time series in all regions of the brain to sort of create this interconnected representation of the brain or brain network. And the appeal of this network approach is that it allows us to relate systemic properties of this sort of communication network in the brain to health and behavioral outcomes of interest. And while functional connectivity analysis obviously underlies network analysis, the subtle distinction between the two is often overlooked in the literature. With the systemic organization of our brains that confer our functional abilities, as we may lose connections due to some adverse health condition. So for instance, you might have an individual, a group of individuals that has a network disconnection, maybe due to a concussion during a sporting event, but compensatory connections may develop to maintain overall organizational consistency and functional performance. And thus you can have different individuals or groups of individuals that have differences in connectivity, but still have the same overall network structure. And additionally, the network approach allows us to not only relate direct brain connections to outcomes of interest, but also allows us to look at indirect connections and their relationship to health outcomes of interest. And hence the need to try to model the entire network simultaneously. And so here's a schematic for how we actually generate our networks from our fMRI time series. So this is when we're creating just a static network or just one network per scanning session. So we get these blood flow levels from cubic subdivisions of the brain over time. And so now we have these time series, and we look to see how these time series are functionally associated with each other. Often we just use simple correlation, but there are many approaches you can use. You can use other association measures like mutual information. You can use modeling approaches, but most people just use correlation. And then the larger the correlation, kind of the thicker the line in this network. And this network here is what becomes our unit of analysis. And so we'd like to be able to, in some sense, relate this network to both sort of endogenous network properties or network features, as well as exogenous covariates of interest. And the field more recently has moved to actually looking at how these networks dynamically change within a scanning session. So instead of creating just one network per scanning session, often we'll create a set of networks per scanning session And often these are calculated by just using sort of like a sliding window correlation, but there are also other approaches here. And in the same sense, we still want to be able to try to relate these sets of now networks to endogenous network features and exogenous covariates of interest. So I'll pause briefly there to see if there are any questions just about the general setup. So once we have our networks, the next step is to try to describe the sort of network properties. And so there are lots of categories of network properties and lots of metrics within each category. But here I just show some of the more popular ones. So functional segregation kind of gives you a sense of how well the network can communicate within itself sort of locally, or local communication. And it's often measured with the metric called the clustering coefficient, which is illustrated here. And the clustering coefficient is just the proportion of a brain region's connections that are also connected to each other. So you can kind of think of it as like in a social network context, it's just a proportion of your friends that are also friends with each other. And then the functional integration kind of gives you more of a sense of the brain's ability to communicate globally. And it's often quantified with the metric called the path length. And the path length between any two brain regions is just the minimum number of edges that have to be traversed to get from one region to another. And then you just average that over all the entire network. And then the resilience of the network is often quantified with the degree distribution. And the degree is just the number of connections a region has. And so certain degree distributions are more resilient to certain types of injuries than others. And then we also look to see how well the brain can efficiently transmit information. And there's several sort of centrality metrics that attempt to capture this. And we often use one just called leverage centrality. And then we also look to see how well the brain could subdivide into sort of these subnetworks for an efficient division of labor. And its ability to do that is often quantified with something called the modularity statistic. And so these are all for metrics for static networks, and sort of descriptive metrics for dynamic or time-varying networks are relatively nascent, but are sort of rapidly being developed. Now, the one thing that is often ignored is that these metrics that we just talked about, these descriptive metrics, are functions of an estimated network. So it's not like in social network analysis where it's pretty much a fixed and known network. But we typically treat our networks here as fixed and known, even though they're estimated. And so these metrics are actually estimates themselves of certain probability distributions. And so more formally, if we denote the true network as Y as a function of nodes and edges, and the estimated network is Y hat, since here we're estimating the edges, and denote the true value of some network metric like clustering coefficient as G of Y, and our observed value as G of Y hat. We're often interested in whether or not our estimate is unbiased, what's its distribution, et cetera. And so there have been a couple of works that have attempted to address this for one metric. So these two groups quantified the propagation of error from network estimation to the density metric, which is just a proportion of the number of existing edges to the number of possible edges. But again, this is just one of many metrics commonly used, and is a relatively simple metric, but it's still very complicated to try to propagate that error through to the network metric and subsequent inference. And so this isn't an area we particularly focus on, but it's important to highlight that this area of network error propagation has barely been tapped and provides an extremely fertile ground for future statistical work. So I'll briefly stop there to see if there are any questions.
So a question about the networks. Are they like weighted directed networks?
So initially, most people use just binary networks, but now most people use weighted undirected networks, because usually with fMRI, we don't have the temporal resolution to really infer direction, at least if we're looking at the entire network.
Then there's like different neurotransmitters, right? Like, do you actually account for that, or is it just signals sort of like you see?
Yeah, it's just a signal, that blood flow.
Okay.
Yeah. Thank you. So now appropriately modeling and drawing inference about the network phenotype relationship. So once you actually have your network, how do you relate that to outcomes of interest? kind of requires the integration of statistical tools with network neuroscience methods, which provides us with a pretty unique challenge. So there have been many approaches adapted and developed. There are sort of traditional network models like ergoms, often used in the social network literature, tensor regression, Bayesian approaches, statistical learning techniques. But few of these integrate these two domains of network neuroscience and statistics, while also allowing for group and individual level assessments. And additionally, the optimal methods are going to vary by your question of interest. And so we really need a suite of complementary approaches. And so we've attempted to sort of add to this suite by developing three frameworks: a mixed modeling framework, a distance regression framework, and a hidden semi-Markov modeling framework. But a lot more work is needed. And I'll touch on these three approaches sort of in the upcoming slides. So the motivation for our mixed modeling framework was we wanted to be able to assess the effects of multiple variables of interest and network features on the overall network structure. So if we denote the network of subject I at time point T during task L is YITL, then we have our covariate information in our parameters. In some sense, we want to be able to try to model the probability density function of the network given those covariates and parameters. And so we developed a two-part mixed modeling approach, which models both the probability of a connection existing between two regions as a function of network metrics and covariates, and then the strength of that connection if it exists in a similar fashion. So in statistical notation, we're just using a logistic mixed model for the presence and modeling that as a function of network metrics, some covariate of interest, confounders and interactions, and then the Fisher-Z transformation on the strengths and modeling that in a similar fashion. And then these theta parameters can contain subject-specific and dyad-specific, where a dyad is just a pair of regions, random effects and random error terms to try to capture some of the dependent structure in the networks. And so far, random effects, we had subject-specific intercepts, we had network metric random effects, we had spatial distance random effects, and then we had these delta parameters, which kind of capture the residual propensity of nodes J and K of the given dyad to be connected and the magnitude of its connection. And we assume variance component structures for the random effects and sort of the standard conditional independence for the error terms. So as an example, we use this approach to examine the impacts of pesticide and nicotine exposures on farm workers' functional brain networks in a farm worker study that we've been a part of for quite a while. And so these two figures here sort of typify the main results that we found, namely that the farm workers' brain networks, and this is at rest, tended to be more modularly organized with this sort of higher functional specificity and lower between module integrity, or these weaker connections between the modules. And so this sort of network architecture at rest has been linked to a number of sort of detrimental health outcomes. And so we can see that this pesticide exposure is likely doing something damaging to overall brain function. And so next we were interested in trying to be able to look at rest to task differences in brain networks and how those are related to health outcomes. So before we were only looking at one network per person at a time, but we wanted to be able to look at multiple networks per person at a time to look at across task differences. So we just introduced this L subscript, where L here now denotes the task, and we introduced that into both models. And then we just updated our random effects. So now we have sets of random effects for each task. And we updated our assumptions. And then we additionally added covariance parameters for each random effect and their counterparts across tasks for each group to try to capture the temporal dependence. And so, for example, in the 2-task-1 group case, we have these covariance matrices for our random effects. And so for this approach, we actually used it to look at some aging data that we were working on, where we were interested in sort of rest to task brain network differences between young adults and older adults. And so again, these figures sort of typify one of the main results that we found with our model, namely that for young adults, when they go from rest to a visual task, their brain sort of snaps into this sort of task efficient architecture, this interconnected sort of high degree, high strength connections of globally efficient hubs. Whereas for older adults, their brains actually tend to not snap into this more task efficient architecture. And these were on average. So obviously you do have some older adults who have young looking brains and some younger adults who have older looking brains. And so we were then interested in trying to extend our framework into the dynamic network context, given that that's where the field was moving and that it provides some additional insight. And so we updated our model by adding these orthonormal polynomial terms in the fixed and random effects. And so we implemented this model with some publicly available data, the Human Connectome Project Young Adult data, where some of our collaborators were looking at the IQ scores and wanted to see if there were brain network differences between those who had higher IQ scores and those who had lower IQ scores in this sort of publicly available data, and looking for dynamic differences. And so basically what we found is that those in the top quartile of IQ scores, dynamic changes in their modularity tended to be more driven by strengthening these within community connections while still maintaining these strong connections between communities, which allows sort of optimal, efficient division of labor while still allowing for information transfer across the entire system. Whereas those within the lower quartile tended to have these weaker within community connections and sort of weaker between community connections, which may lend itself to less optimal information transfer.
Sean, what's the difference between the left to the right?
So here we're looking at dynamic brain network changes. So changes within a scanning session. So this just gives an example of how these brains when they do become more modular, those in a higher quartile, they become more modular by actually strengthening within, whereas these become more modular by kind of weakening between connections in these dynamic networks.
Is the overall edge strength constant?
So the overall edge strength is typically not constant dynamically. I think it can change dynamically. Yes.
May I ask you what's the size of the sphere?
So that is the number or the strength of its connections. So the larger the sphere, that means the sum of its connections are larger.
Is it measured as degree?
Yeah, right. Degree. It's like weighted degree, basically. Yeah.
Okay, question. Can you go a little bit back?
Yeah.
To the model one. This one. More like a basic question. Do you add some constraints for this PIJK? For example, the negative, the definitely positive, or something like. At least I see for me some kind of constraint need to be see this PIJK. For example, the PIJK should be positive definite, and the summation of this one for each I, the JK should sum up to 1, and it's more like compositional data, or it has some kind of spatial constraint.
Right. So the only constraint, so I didn't touch on too much. So when we actually create our networks, we often threshold them. So it used to be early on in sort of the network literature, people would do binary networks threshold using a thresholding technique and just say 1 if they think there's a connection, 0 if not. Now we're doing weighted networks, but we still typically threshold at 0. So these are all anything below 0 is considered a non-connection because there's a lot of work that says negative, especially negative correlations from imaging are typically artifacts. And so they typically just cut off those. And so a 0 would be kind of a non-connection, and then it's weighted up, 0 up to 1, basically. And so we developed a MATLAB GUI interface that implements these mixed modeling approaches, and it interfaces with SAS, R, and Python. And so for future work, what we're doing is we're looking to sort of integrate our multitask and dynamic modeling framework so that we can look for cross-task differences in dynamic network changes. That's we haven't gotten started on that yet. So I'll briefly pause before I get into the next.
For the dynamic one, do you add some kind of constraint for the different time point?
For the polynomial, well, there you more have to you want to still have a parsimonious model. So so we just kind of assess like we use information criteria and other approaches to try to assess the proper number of polynomial terms. So the motivation for the distance regression framework was that we were interested in trying to assess the spatial, topological, and distributional consistency of key node sets. And key node sets are just brain regions that you think are important. So that can be informed by your sort of question of interest. Here, for example, we often use just the top 20% highest degree nodes or most connected nodes. Those are called just hub nodes in the literature. So if your key nodes are hub nodes, for example, and you are interested in seeing whether or not those hub nodes are in different spatial locations in two groups, the question is, how would you do that? So here we can see that for these two groups, they appear to be in different locations if you look at these overlap maps, but we wanted to have a way to try to formally assess this. And so we developed an approach to doing this that also allows us to control for confounding variables. And so we developed this sort of distance regression framework, where here we're looking at the distance between the networks of subjects I and J. It's a function of differences in confounding variables and differences in some covariate of interest. And so, for example, if you wanted to assess whether or not there's a relationship between the spatial location of hub nodes and IQ, in this setup, you would use what's known as the Jacquard index for your distance, as it gives the proportion of overlap of these hub nodes, and assess the null hypothesis that beta 2 is equal to 0 in this setup if you're also looking to control for age, sex, and treatment group.
So what is the distribution of EIJ? Does this depend on the distance IJ's choice?
It does to some extent depend on the distance DIJ choice.
How do you do this assumption?
Right. So I'm kind of getting to that because that is where we run into some issues, because you can't necessarily just use a standard F test. So I'll kind of talk about that a little bit. It's also important to know that here what we're doing is we're summing across the rows of the connectivity matrix to get these nodal degree vectors. So we often subdivide our brain into 268 brain regions. So here we're just getting this degree vector and then calculating the distance or similarity between the nodal degree vectors. And so we assess several distance metrics, but here I'll just touch on three. So one was just the Euclidean distance, which is obviously location specific. One was the Kolmogorov-Smirnov statistic, which is not location specific. So here we'd be looking for distributional differences. And then again, as I noted before, the Jacquard index, which just gives the proportion of overlap. So as already noted, the errors are what kind of become the problem in this setup, and that renders sort of the standard F test inappropriate. More so because of the correlation pattern than necessarily the distribution, because if you remember, we're expanding our N subjects of data into N choose 2 pairwise comparisons. And so obviously the Jacquard index between subjects 1 and 2 is going to be correlated to that between subjects 1 and 3. And so we assessed several different estimation and inferential approaches within this framework. So we looked at, again, the standard F test just for comparison, but we figured it wouldn't have good properties given the correlated errors. We looked at what we just called an F test with individual level fixed effects, or F test with ILE. And we hoped that by accounting for individual level effects, we could induce independence in the errors and possibly render the F test appropriate. We looked at a feasible generalized least squares approach, which I just kind of think of as sort of a mixed model light. And here we have to estimate the covariance matrix of the errors, and we use the trick to do that, but I'll leave that out here for the sake of brevity. And then we use a permutation approach where we just permuted the residuals of the model. And we compared our approach to the multivariate distance matrix regression, or MDMR, as this is probably the most common approach for these types of analyses. And we compared our approach to their permutation and analytic p-value method. So for our simulations, we had 100 subjects. We had 2 categorical variables, sex and treatment group, and then 2 continuous variables, IQ and age. And then we just labeled them this way for purposes of explication. And so we did 4 sets of simulations. So 3 of the simulations were location specific, and those are shown here. And 1 was more distributional and non-location specific, and I've left that one out. And so here what we have is we simulated 1 connectivity matrix for each subject, 268 nodes. So this is 268 by 268. And then this teal region is just pure noise. The yellow region is where everyone has the same amount of signal. And then the purple region is where signal depends upon covariance, IQ and treatment group in this case. And so here we have the power results for simulation 1, and the power results for the other simulations were pretty similar. And as we expected, which is not shown here, the F-test did not control type 1 error, but all other approaches did control type 1 error. And we can see that our approach, particularly the F-test with ILE, actually far outperforms MDMR in terms of power for the KS statistic, and is comparable to MDMR for the Jaccard and Euclidean. And so we implemented our approach to, again, look at human connectome project young adult data. And for these data, we started with 1,200 subjects. We're again looking at fluid intelligence scores. We controlled for these confounders. And after we removed kind of subjects for missing data or due to quality control, and then just selected 1 person from each family, we were left with 397 participants that had resting state and working memory connectivity matrix data. And so one interesting result of note was that we found that the spatial location of those hub nodes or highly connected nodes were in different locations in those in the bottom and top quartiles. So for those in the bottom quartile, their hub nodes tended to be in this area called the default mode network, more so than for those in the top quartile. And then when those in the top quartile were engaged in a working memory task, their hub nodes tended to migrate more towards what's called the central executive network, whereas those in the bottom quartile, their hub nodes tended to somewhat stay stuck in the default mode network. So next, our collaborator was interested in analyzing the retest data from the Human Connectome project. So it has a setup here where each person is scanned under 3 different tasks and under 4 repetitions per task. And so we wanted to be able to extend our framework from just kind of the single network context to now be able to handle this sort of multitask and multi-session data. And so what we did before, we were looking at those sort of nodal degree vectors, comparing distances between those nodal degree vectors. Whereas here, we sort of switched into now we're looking at distances between sort of the whole connectivity matrix, which kind of changes our focus from nodal degrees now to edge weights. And so to extend our framework to this context, we recently extended the approach to now where we're looking at the distance between the networks of subjects A and D for task B for all repetitions C and F. And so again, if we wanted to now look at whether or not the spatial location of now the 20% strongest edges differs by IQ after controlling for age, sex, and treatment group, we'd have the following model using the Jaccard again as our distance metric, and again, assessing the null hypothesis that beta 2 is equal to 0. So in addition to the distance metrics we used before, we examined 2 additional metrics for this modeling development. So we looked at the log Euclidean Riemannian metric, which we'll just call LERM, as it's been shown to capture several types of sort of location-specific differences as well. And we just looked at the standard Pearson correlation distance. And we, again, included the standard F-test for comparison, and then looked at two new sort of estimation and inferential approaches. So one that we just called the standard F-test with now scan level fixed effects, which is analogous to the previous F-test with individual level fixed effects. And here the scan variable is just a known indicator variable for the brain scan of individual eye during task J and repetition corresponding parameter. And then we proposed what we called our 3-Bantor model or mixed model for multitask brain network progression. And so we added on to the scan ID variable and now have these ID variables where they're indicated variables for comparisons between distinct individuals A and D on the same task B with this corresponding random effect. And so we had the same simulation set up as before to sort of compare our approach in this multi-session, multitask context, again, to MDMR. But we had to use a sort of novel strategy to simulate semi-positive definite matrices so we could test the metrics on the Riemannian manifold. And I've left that out here too. So we had the similar simulation set up as before, except now we're simulating 12 connectivity matrices for each subject instead of just one. And again, the teal is just noise. The yellow is where everyone has the same signal. And the purple is where signal depends upon covariance. And I just show you this here to show you that we had to think about how we wanted to simulate the correlations in our simulation study. And so we ended up basically simulating a within task correlation of 0.7 and a within repetition correlation of 0.3. And so here I show the results from the task 1 simulations, and the results again were pretty similar for the other simulations. And so here we could see, you don't have to pay too much attention to the details, but we're looking at both type 1 error in power and then type 1 error in power. And basically the take-home message was that our 3-Bantor method was actually the only method that controlled type 1 error across all of the metrics. And it had more power than all of the other approaches when those other approaches did control the type 1 error. So we felt confident in moving forward with our approach. And so for kind of current and future work, what we're looking to do is be able to extend this framework to be able to handle familial correlation. Because if you remember before, I was saying we had to randomly select one individual from each family. But in the Human Connectome Project data and other publicly available data like the ABCD study and others, many of them have lots of familial correlation. So here we have 450 families and 269 twins. And so we're looking to extend our framework to be able to handle that familial correlation. And so we're still in the early stages of this, but what we're proposing right now is something that's called MLM-Bantor, multi-level brain network regression. And we're basically adding these fam and twin variables that we're using in conjunction with both random intercepts and random slopes. Here we just have the random intercepts. We're looking at random intercepts and random slopes for these variables and trying to artfully construct them to be able to account for the familial correlation. And so I'll briefly show you some preliminary simulations on our current construction of them, but we're looking at other constructions. So I won't get into the details of the actual construction here, but just sort of give you some takeaways from some preliminary analyses we're doing with our current construction. And so again, for these preliminary simulations, we're simulating about 100 to 105 subjects, 2 to 6 siblings per family, and 1 twin pair. We have this sort of basically the same setup as before with the noise and the signal. And so here we have results from some of our preliminary simulations. And basically, I'll just more or less highlight this right side. because we had to kind of think about how do we simulate these correlations or realistically. And so we've looked at basically kind of 6 sets of correlations, both looking at within family and within twin covariate correlation, and within family and within twin time series correlation. Because remember, we use those time series to create the networks. So we wanted to induce correlations in many ways. So here is just what we would get if we assumed everything was independent. But basically, you can see we've kind of been looking at different kind of levels so far. And sort of the takeaway message we've gotten so far is that our MLM Bantor method with the current construction appears to be working fairly well, but we're still trying to make some tweaks to see if we can make it perform better before we sort of proceed with it. And once we do kind of have our sort of final model, we're planning to revisit the Human Connectome project data, because the biggest change will be now we'll be able to actually probably use about 819 of the participants instead of just 397, since we can include people from the same family. So I'll pause here before I get into the last few slides, see if there are any questions, comments. Yes.
Very interesting method. I'm wondering how can we use this method for longitudinal data to measure the longitudinal change of 2 time points?
Right. So that is something that we're also thinking about. Actually, I could have put that in here as a future direction. It can't currently do that, but we're starting to try to think of some ways we could extend it in the longitudinal direction too, because we do have some longitudinal data we're interested in analyzing, but that we haven't really thought through completely, but it's on our radar, definitely. So,
may I ask a second question?
yes, sure.
So you mentioned that you use the retest the data set, right?
Right.
So if we measure the age effect or the effect of other factors, are they reliable across the scans? Can you detect the reliable effects across the different scans?
To some extent, the problem with there is that the sample size is so small, we don't really trust it. But so I think what we could use there, the sample size isn't large enough for us to really be able to have faith in what we see, I guess.
Okay, sure. Okay, thank you.
So this sort of last framework is actually work spearheaded by my colleague Heather Schoppel, and she sort of joined us about 2020, and we started sort of working on these hidden semi-Markov modeling things together. But this is more sort of her work, whereas I'm more focused on the first 2 frameworks. But for this approach, basically, we're assuming that the unobserved states, and so here we're looking at dynamic dynamic network state changes within a scanning session. And so we make the assumption that the unobserved states, the S's, evolve according to some semi-Markov process that's characterized by these state transitions, the red arrow, so going from state 2 to state 3 or state 5 to state 6, and sojourn distributions, which is just a time spent in a state, and those are those dashed lines here. And this is what we observe, the observed kind of imaging data. And so here's how the kind of approach is defined. So if we assume that the bold measurements or the imaging measurements of the P-read brain regions of interest at the T time point for the ith participant are in YIT, and again, the S's sort of index the hidden states. We assume that the YITs follow a p-varied normal distribution with some mean mu and some covariance matrix, which you can think of actually as our network sigma. And so each state has its own mean and own network. And then we can then use a complete data log likelihood to obtain group level estimates of network structure, transition probabilities, and sojourn distributions for each state, as well as to conduct inference about these various estimates. So for example, in one analysis where she was looking at the difference in dynamic network state changes between children with ADHD and then typically developing kids. She found two states that were significantly different between the two groups. And these are just their network matrices or adjacency matrices for those two states. And so what she found was that those who had ADHD tended to have a higher probability of spending less time in state 3, and vice versa for the state 6. Those typically developing kids had a higher probability of spending less time in this state 6. And so for future work, she's looking to develop a mixed effects version of this model for estimating both individual and group brain network dynamics. She's looking to extend this to be able to handle more spatially resolved networks. So networks where we can look at greater than 500 regions at a time. And also she's looking at a way to try to determine the optimal number of states to fit into the data, because often we just do that in an ad hoc fashion. And so in conclusion, network analyses may revolutionize our understanding of brain function, but we need to keep developing methods to keep pace with the questions of interest. And given the variety of scientific questions, we definitely need a suite of tools. And so we've attempted to add to that suite here. And again, more attention needs to be paid to error propagation from network estimation. So thank you for your time.